Today we're going to talk about Crispr-Cas9. When inserted into cells, Crispr-Cas9 binds to a specific sequence and makes double-strand breaks. Specific binding is assisted by two components. PAM sites are three nucleotide sequences in the genome found downstream of the target sequence. The specificity of the PAM sites, along with the homologous targeting by the guide RNA, allows for Crispr-Cas9 genome editing. The second specificity component is the guide RNA of CRISPR. The guide RNA is composed of CRISPR RNA and transactivating CRISPR RNA, which is also called the tracer RNA. The CRISPR RNA is about 17 to 20 nucleotides long and is homologous to the target sequence of the genome. The CRISPR RNA binds to its homologous target and forms a double-stranded DNA-RNA hybrid. The tracer RNA binds a CRISPR RNA to the Cas9 nuclease. Cas9 has two nuclease activities, one to cut the DNA-RNA hybrid and one to cut the remaining single-stranded DNA. This creates a double-strand break which can be recognized by repair machinery.
If the cell repairs the double-strand break with non-homologous end joining, it will add or delete a few base pairs and normal repair machinery will join the strands back together. This often induces a missense mutation, which allows scientists to knock down specific genes. Another way the cell can repair double-strand breaks is through homologous recombination. If a DNA template is provided, DNA polymerase II can make specific edits and insertions into the genome. Crispr-Cas9 can also be used for fluorescent localization of genomic sequences. If the Cas9 nuclease is deactivated, the guide RNA of CRISPR can be used to localize specific loci in the genome. A GFP tag is added to the CRISPR complex and fluoresces when binding occurs.
CRISPR has the potential to be used in gene drives. A gene drive, as defined in Science magazine, is the practice of stimulating biased inheritance of particular genes to alter entire populations. This occurs naturally whenever selfish alleles cause their own inheritance to be favored over that of their homologous allele. This is usually enacted by endonuclease genes, which specifically cut the homologous allele, causing homologous recombination to repair the double-strand break using the selfish allele as the template. If this occurs in germ cells, the frequency of the selfish allele increases in the next generation. Previous attempts at developing gene drives have been limited by the lack of gene-specific endonucleases. Because CRISPR allows directed endonuclease activity at a site determined by the researcher, it could allow the use of any gene or combination of genes in a gene drive. Just this year, gene drives have been demonstrated in the lab in yeast and Drosophila. Importantly, these gene drives have also been proven reversible. The possibilities of such a technology are monumental. It could be used to make mosquitoes unable to harbor malaria and dengue fever, to reverse pesticide and herbicide resistance in agricultural pests and weeds, or to eliminate the entire interbreeding population of an organism. Of course, scientists have called for the utmost caution and stringent regulation in the use of gene drives in natural settings.